welcome again to Let's Talk Forensics with Pastor Dan. We're in a different environment here today. The roads are a little bit rough outside, so I'm doing this at home. And we're going to do DNA extraction lab today. And it's not going to be electrophoresis. That's really expensive. Most of you aren't going to get a chance to have one of those. It takes way too long too. So what you're going to do is you're going to need 3 items if you can get them. All right, first thing you need is a bottle of Gatorade and a small Dixie cup. Second item you're going to need is soapy water. The third item you're going to need, if you can get it, is ethanol. That's like grain alcohol. If you can't do that, isopropyl alcohol probably work the same. So what you're going to do is take some of the Gatorade here, pour it into a small Dixie cup, put in your mouth, swish it back and forth. Remember we talked about before extracting off the cheek cells? So that's exactly what we're going to do. We're going to spit that out. Now, I would always pour that into a test tube. You may not have a test tube, just some small vial that's clear, or maybe a really small glass that you can start to see some things in there. You're then going to take the soapy water. The soapy water is going to act as a limiter, and it's also going to begin to pull the DNA out of the Gatorade fluid. And then you're going to have to do what is called an amplify process. Now, the amplify process we're going to do is real simple. So if you can take that container, make sure it's small, put it underneath your armpit. And it says here, if you look right here, you're going to do it there for 15 minutes. And the heating process is what pulls the DNA out of the soap and makes it more combined. And then after that, you should have inside of there, add a little bit of the alcohol. It'll make a secondary fluid, an ethanol layer. In that layer, you should see a little small, either white dots or a white mass if you have a lot of it. And that then is your DNA. So that's the process. You can see. Go ahead and look at that over there. The only observation is, what does it look like? It's going to be a mass, is what it looks like. And then what is added to the solution in the Lysol process. So what do we act to begin to pull that out? Think about that. And then, of course, your conclusion. So that is your DNA lab. Hopefully not too bad. Take care. God bless. Merry Christmas. Bye.